Hello everyone. Today, we're diving into a fascinating topic, the axolotl paradox. We'll explore how this remarkable species, teetering on the brink of extinction, may hold invaluable biological keys to unlocking truly immortal health. The axolotl presents a stark duality, ubiquitous in labs yet tragically vanished from the wild. This species, scientifically known as Ambystoma mexicanum, is critically endangered. Its wild population in Xochimilco, Mexico, has collapsed dramatically, as shown by the graph on the left, plummeting from hundreds of thousands to near zero. Yet, simultaneously, a thriving global population exists in captivity, with millions in labs and the pet trade, indicating a rapid increase. It's a paradox, a species both abundant and functionally extinct. The tragedy of Xochimilco isn't due to poaching, but an environmental catastrophe. Three main factors contribute to this decline. First, invasive species. Government-introduced carp and tilapia consume axolotl eggs and compete for resources. Second, urban pressure. Severe pollution and water quality degradation from Mexico City's urbanization have taken a heavy toll. And third, habitat loss. The canal system itself has been drastically reduced. It's crucial to understand that the pet axolotl we see today is a distinct, captive-bred lineage, serving as a biological ark for a species that is almost certainly functionally extinct in nature. The axolotl is truly the Peter Pan of the amphibian world. Unlike a standard amphibian, which undergoes a complete metamorphosis from egg to larva to an adult that lives on land, the axolotl exhibits a phenomenon called Neoteny. This means it reaches sexual maturity while retaining its larval features, staying in the water and keeping its gills. This is due to a low-activity hypothalamo-pituitary thyroid axis. This permanent larval state is incredibly relevant to our discussion, as it's directly linked to their high cellular plasticity, which forms the very foundation of their remarkable regenerative power. This regenerative power allows them to build what appears to be the perfect beast. When an axolotl loses a limb, the regeneration timeline is astonishing. On day 0, after amputation, by day 10, a blastema forms a mass of indifferentiated cells. By day 30, these cells begin to redifferentiate and grow, leading to a perfect replica by day 90, with absolutely no scarring. This stands in stark contrast to mammalian healing, which typically results in fibrosis, or scarring, creating stiff, non-functional tissue. The axolotl's blastema formation, however, ensures perfect tissue architecture. This leads us to a critical question: if their cells can proliferate so rapidly to grow an entire new leg, why don't they grow out of control into tumors? This brings us to the cancer anomaly. Axolotls exhibit an exceptionally high resistance to cancer. The incidence of malignant tumors is a mere 1.8%, even when chemically induced. They are not entirely immune, but they are highly resistant. This resistance manifests in two ways. Young axolotls are often regressors, meaning they can spontaneously shrink tumors. Adults are rejectors, often failing to develop tumors entirely despite exposure to carcinogens. As Laguna's Rangel noted in 2026, increasing evidence suggests that axolotls display robust resistance to cancer, making them a valuable model for tumor suppression. The axolotl's cancer resistance isn't attributable to a single factor, it's a multi-layered defense system. It's a conspiracy of physiological, cellular, and molecular traits working in concert. We can break this down into 4 key pillars: their unique physiology, which acts as a metabolic shield; their genetics, forming a genomic fortress; their biochemistry, an insulin brake; and their immunology, featuring an innate patrol system. Let's start with the metabolic shield. Cancer cells notoriously rely on the Warburg effect, requiring massive energy and glucose to fuel their rapid growth. However, the axolotl's cold, low-energy environment restricts these resources. Their metabolic rate, compared to mammals, is significantly lower, operating at 15 to 20 degrees Celsius versus 37 degrees. This creates a hostile environment for tumorigenesis, making it physically difficult for cancer to rev up and proliferate, essentially starving the tumor from the outset. Further, starving the enemy, axolotls possess a unique biochemical trait: low insulin sensitivity. They naturally have lower blood glucose levels and reduced sensitivity to insulin compared to many other species. By keeping these mitogenic, or growth, signals dampened, they effectively reduce the availability of the very growth factors that cancer cells typically hijack to spread and proliferate. This acts as an internal brake on uncontrolled cellular expansion. Next, we examine the 32 gigabit fortress, their genome. While the human genome contains 3.2 billion base pairs, the axolotl genome is a colossal 32 billion base pairs. This massive genome is not just larger, it's structured differently, filled with extensive introns, which are 25 times longer than in humans, and numerous repetitive elements. This vast amount of non-coding DNA acts as what we call mutation buffers. Statistically, any harmful mutation is far more likely to land in this junk DNA rather than disrupting essential coding genes, thereby protecting them from genetic damage that could lead to cancer. The axolotl also boasts a formidable innate patrol within its immune system. Natural killer precursor cells, or NCCs, are the hunters in this system. They actively bind to and destroy tumor cells using specific receptors and lytic granules, effectively eliminating cancerous threats. What's even more remarkable is their ability to regenerate their thymus. Unlike humans, whose thymus shrinks with age, leading to immune decline, the axolotl continuously regenerates this vital organ, maintaining robust immune surveillance throughout its entire lifespan, effectively preventing immune aging. Beyond these, axolotls engage in a form of internal chemical warfare. Extracts from axolotl oocytes, or eggs, have been shown to reprogram cancer cells. These extracts can strip away epigenetic markers that silence tumor suppressors, effectively reactivating the body's natural defenses. Furthermore, their skin mucus is a powerful source of antimicrobial peptides. These compounds have been found to upregulate tumor suppressor genes and down-regulate oncogenes. The key insight here is that the axolotl body naturally produces compounds that function like targeted chemotherapy, attacking cancer at its molecular roots. It's important to understand that regeneration is, in many ways, the opposite of cancer. Cancer, or malignancy, involves uncontrolled dedifferentiation, leading to a chaotic structure and a stiff, fibrotic environment. In contrast, regeneration, particularly through the blastema, involves controlled dedifferentiation, maintains positional memory, and creates a soft, scar-free environment. The regenerative microenvironment of the blastema actively suppresses malignancy. It compels cells to differentiate and get to work repairing tissue in an organized fashion, rather than growing selfishly and chaotically as cancer cells do. So, how can we apply this knowledge to cracking the code for human health? We cannot become axolotls, but we can certainly mimic them. We can synthesize peptides found in their skin mucus, which have anti-cancer properties. We can also explore ways to mimic their metabolic slowdown to help manage tumor growth in humans. Furthermore, understanding how they induce scar-free healing could prevent the fibrosis that often supports cancer development. The urgent challenge now is to understand the biology of this extraordinary species, which is tragically disappearing from our physical world. To summarize, the axolotl's survival toolkit against cancer is multifaceted. First, their metabolic shield cold body temperature and a low metabolic rate starve energy-hungry tumors. Second, biochemically low insulin sensitivity reduces growth factor signaling, which cancer cells exploit. Third, genomically their massive non-coding DNA regions act as buffers against harmful mutations. Fourth, immunologically natural killer cells actively hunt tumors, and their ability to regenerate the thymus prevents immune aging. Finally, regeneratively the blastema environment enforces cellular order and actively prevents fibrosis, which can otherwise promote cancer. Here are the sources and data that informed this presentation. Our primary reference is a forthcoming paper by Lagunas Rangel, titled, "Are Axolotls Resistant to Cancer? Possible Explanations," slated for 2026. We also drew insights from community discussions on Roxolotls on Reddit. All visuals have been adapted from Lagunas Rangel's work in public domain biological databases. Thank you.